So today we have Dr. Ramirez, Julio Ramirez, Chief Research Scientist for the Norton Infectious Diseases Institute, will be giving us an update on what is happening with the COVID vaccines. So thank you, Dr. Ramirez.
Okay, thank you, Ruth. Thank you for your attendance. The idea is to give you an overview of the COVID-19 vaccines, a little bit of the worldwide perspective of what happened with the COVID-19 vaccines. And to give you this overview, I'm going to have this in the next 30 minutes. We're going to discuss these 5 points. First, what is the immune response to the natural infection, to natural COVID-19 infection? And in this regard, we know that this virus I represent here, this is an RNA virus, the SARS-CoV-2, that attach to our epithelial cells using the receptor, the angiotensin converting enzyme 2. This virus has the RNA, and here in this RNA have all the genes that produce the proteins. Here I represent the virus has 29 proteins. It's able to produce, they have 29 genes. The gene of interest for us here in red is going to be the gene that is going to produce the S protein, the spike glycoprotein. This gene is going to produce approximately 1,300 amino acids. And this is the gene for the spike protein. And the spike protein is the one that the virus used to attach to our cells. Once the virus attaches to our cell, they penetrates the cells, endocytosis, release of the RNA into the cytoplasm. This RNA, 2 things happen. The RNA is going to multiply, produce multiple copies of the RNA, and also this RNA is going to transform into the messenger RNA. And then this is the way that the genetic information goes to our ribosomes, and then we are going to produce, our cells are going to produce the viral proteins. An important component is the viral proteins that cut the proteins at the proper size. And then we are going to produce the viruses. And there's going to be multiple viruses released. Now, what we are, once the virus is multiplied inside of our body, our antigen-presenting cells, that primarily, if the virus is in the lung, are going to be the alveolar macrophages, or it may be the multiple cells that are going to capture the viral antigens, are going to present the antigens to our immune cells. And our immune cells, B cells, plasma cells, go to memory B cells, and then also the T cells. We know that the plasma cells, B cells, are going to produce immunoglobulins, and then we are going to produce a lot of cytokines. And these immunoglobulins are going to be the antibody response. The important antibodies are going to be what we call the neutralizing antibodies. And then they're called neutralizing antibodies because they are going to be able to attach to the spike protein. Then here we see a virus covered with the neutralizing antibodies. Then this virus is not going to be able to attach to any other new cells. Another important consideration of the immune response to COVID is what happened with the T cells, primarily with the T cytotoxic cells, because these cells are going to define whenever we see one of our own cells infected with the virus, it's going to destroy the cells. And this is another way to have a mechanism to eliminate the infection. Okay, then this has been a quick review of our immune response to the natural infection. Then we know today that with a natural infection, we have a good response because we have response of the B cells and response of the T cells, and natural infection may prevent future infections or may make the next infection less severe. Now let's go to the vaccines for COVID. And there are 2 primary types of vaccines for COVID. Then one is the what's called protein-based vaccine. The idea here is that this is the traditional approach to vaccinology in adults. We give the antigen, we inject the antigen to the patient. The most traditional approach for this is in the case of viral infection. Here I represent this, for instance, the SARS-CoV-2 virus. I inactivate the virus. And a way to, some people say, well, I kill the virus, because essentially I have some, at first I grow the virus in cells, and then in the cell culture, I put some chemical that's going to inactivate the genetic material. Then this virus is no longer able to multiply in our cells because the RNA is destroyed. But all the proteins on the surface of the virus are still there. This is what I inject to the patient. Then what I have here is an inactivated whole virus vaccine. These were one of the first vaccines, and these vaccines were produced, for instance, the vaccine that's called Covilo from Sinopharm in China, the Coronavax, Sinovac, another Chinese company, the Covaxin that is produced in India. All these vaccines have been heavily used worldwide. And then in France, there is Valneva, a company that produced the Valneva 2001. And this Valneva inactivated virus vaccine has been approved in the UK. I think it was just 1 or 2 weeks ago, was approved in the UK. Is expected approval in the European Union. And this is the traditional vaccine that I can have the vaccine in the refrigerator and I just give it to the patient. And again, it's similar to what happened, for instance, with the influenza vaccine. Moving along, if I want to inject the protein, another possibility, as we do with, for instance, with the hepatitis B vaccine or the human papillomavirus vaccine, in this case, I get the RNA of the virus and I move the RNA to DNA. And I extract the DNA of a cell. This will be a bacteria, this could be a yeast cell, for instance, is used a lot, yeast. I get the DNA and I recombine the DNA of the virus into this DNA. Then I have a DNA of this cell, maybe a plant cell, an yeast cell, in this case, a yeast cell, and I give it back to a yeast. Then in the laboratory, this cell, when the DNA goes to messenger RNA, is going to produce the spike protein. Then I have a culture of a spike protein that this is what I'm going to inject to the patient. These are the recombinant protein vaccines. And for these ones, there is the Sanofi GlaxoSmithKline in France, Bidpreptin. I mean, this COVID vaccine has some names that are difficult. I don't know why the train names are so difficult, but again, the Sanofi vaccine is a protein-based vaccine, is a more standard approach, and this vaccine, again, these are all finished phase 3, and they are already for different approvals, and the Sanofi vaccine is pending approval for the European Union. The Corbevax, this is the vaccine that was initially developed at the children's hospitals in Texas. This is Baylor College of Medicine that now is interacting with a company in India for mass production. This vaccine was already approved in India. And this is the system that the Finland Institute, that is the Vaccine Institute in Cuba, developed the Soberana 2 vaccine. This vaccine has been approved only in Cuba and Iran. But again, this is the concept of recombinant protein vaccines, with some vaccines already approved. Using the same model of recombinant protein, meaning that you just, there is a cell that produces the spike protein. A little more sophisticated is I have the spike protein and I develop something that looks like a virus. Then in the laboratory using nanoparticles, I develop the structure of a virus, and then I attach the spike protein to these virus-like particles. Then this is the virus-like particles vaccine. And in this regard, we have this is the Novavax vaccine called Covovax, originally produced in the United States. There is the Covovax vaccine. The the COVID fence is produced in Canada, was approved in Canada, this viral-like protein vaccine, and there's another company in China. Then there's a lot of companies and a lot of vaccines approved in multiple countries that are the protein-based vaccines. Since these vaccines usually, primarily the inactivated whole virus vaccine, are low cost to produce, and they are maintained in refrigerators. These protein-based vaccines are going to be the ones that are going to be the primary vaccines to vaccinate in low-income countries, and primarily because they are low cost and doesn't require very low temperatures. Now let's move to the gene-based vaccines. And here the idea is that instead of giving the protein of the virus, I give the gene of the virus to the person, and our own cells are going to produce the protein. Then in the genes-based vaccine, we go back to the gene that is of interest, is the gene of the spike protein. This gene with all these amino acids. Then once I know what are the structure of the gene, then I can try to inject the gene to a person. Now, all here we have the viral vector vaccines. Then the viral vector vaccines, I get the RNA of the virus, these are RNA virus, and move it to DNA. And this DNA, I injected or I incorporated into a virus. This is going to be the vector. Then essentially what I inject to the patient in the vaccine is essentially a virus that has the genetic material of the SARS-CoV-2. The virus goes into our cells. The virus put the DNA into our nucleus. Then in our nucleus, I have the DNA of the SARS-CoV-2. The DNA goes to messenger RNA. The messenger RNA leaves the nucleus, goes to the cytoplasm and look for the ribosomes, and our own ribosomes produce the spike protein. And then our antigen-presented cells is going to recognize this a foreign protein, and we are going to develop the neutralizing antibodies. Then this technology is the vaccine, the trade name JCOV, then the Johnson & Johnson vaccine that use the adenovirus 26. The Convidecia, the CanSinoBiologic, this is the Chinese vaccine that is adenovirus, that uses adenovirus 5. The Russian vaccine, the Sputnik 5, the Gamaleya Institute, that is 2 doses with adenovirus 26 first dose and adenovirus 5 second dose. And this is the Oxford University AstraZeneca, that essentially what the Covishield. But for this vaccine, instead of using a human virus, as the other companies use adenoviruses, this is a chimpanzee virus. This is a non-human virus that you incorporate the genetic material of SARS-CoV-2, and this is what you inject to the patient. Then there are and these vaccines have been heavily used in multiple countries all over the world. Then this will be the viral vector vaccines, injecting the DNA of the virus. Now, there's another consideration is I get the RNA of the virus, I move it to DNA. With this DNA that have the spike protein gene, now transformed into a DNA, I create a plasmid, a silico DNA, and this is what I'm going to inject to the patient. The DNA of the directed DNA without any viral vector. This is a vaccine that then the DNA goes to our nucleus, move to RNA, go to a ribosome, and then we're going to produce the spike protein. This is the Inovio company. The Inno 4800 is the vaccine. They finished phase 3 studies. This is one of the few. This vaccine, for instance, phase 3 studies, this is a vaccine that is at room temperature. They don't even need a refrigerator. These are all new technology, have never been tested before. And finally, we have the messenger RNA vaccines that here you get the gene of the spike protein. This the RNA, put it into a messenger RNA, inject the messenger RNA, go directly to our ribosomes to produce the spike protein. And these are the traditional ones here, primarily used in the US, the Pfizer, biotech, Comirnaty, and the Spikevax, the Moderna NIH vaccines. And then essentially we run now at this moment there are more than 100 COVID vaccines in development. I discussed here the one that's been approved, that finished phase 3. And then we have the protein-based vaccines that are inactivated vaccines, the recombinant protein vaccine, the viralized particle, and then the 3 type of platforms for gene-based vaccines. This is the viral vector, the DNA vaccines, and the RNA vaccine. And this is a brief review of the vaccines. Now let's move to the other topic that I want to discuss is then, what is the immune response to these vaccines? Now, and we go for most of the vaccines, most of these vaccines require 2 doses of the vaccine, usually 3 to 4 weeks separation. And the goal is that I give the 2 doses of the vaccines, and then if the person is going to have an optimal immune response, we are going to be in this range here, that is when you have the neutralizing antibody levels, meaning that you have enough antibodies that the virus is going to want to get in, the antibodies are going to attach to the spike protein, and the virus is not going to be able to infect our cells, and we are not going to develop infection. One of the challenges is that after we get the vaccine, when we, some of our patients, well, I want to get my antibody levels. Well, the antibody level is the level that you have of IgG after vaccine, but the antibody levels doesn't tell you how well the antibodies are going to attach to the virus. For this, you need to have neutralizing antibody levels, and the neutralizing levels that are performed only in research settings. Then if someone say, well, I want to see how well protected I am after my vaccine, essentially it's not recommended to get antibody levels because we don't measure neutralizing antibodies. Now, one thing that we know is that this will be the optimal immune response. You get a level that you're going to be protected. Now, some patients or some individuals don't get into the optimal level, they get a suboptimal level, and some individuals after the vaccine, they don't get any immune response. Then we know that no immune response, this is usually in the patient that is immunocompromised or the severely immunocompromised. And this is an important consideration, not just for the SARS-CoV-2 vaccine, but for other vaccines. Then, knowing this, that this happened during the initial phase 3 studies, the concept now is that if we are going to give any type of COVID-19 vaccine to an immunocompromised individual, the primary series of the vaccine are going to be 3 doses. Then 1 dose, 3-4 weeks, second dose, 3-4 weeks, another dose. Then for the non-immunocompromised, usually the series is 2 doses. For the immunocompromised individual is 3rd dose, because with the 3rd dose, we are going to have more patients immunocompromised that are going to be able to get into the optimal immune response. Still, even with 3rd doses, some patients that are immunocompromised, they will not get into the optimal immune response. And this will be the immune response to the vaccines. Now, going back to continue with the immune response to a vaccine, you see here that the optimal immune response, I go back to this here. This is, let's say, 6 months after the vaccine. What happened 9 months, 1 year after the vaccine? I have this line that significant number of the population, they have an optimal immune response. Now the immune response started to wane off and they have now suboptimal immune response. When we learned this, that even the the non-immunocompromised individual, after several months, the amount of neutralizing antibodies start decreasing. This is when we develop the concept of a booster dose. Then at this moment, try to optimize the immune response for anyone. Either you receive 2 doses or 3 doses after 6 to 9 months, then we're looking at the booster dose. Okay, now let's look at how do we understand now what happened with the immune response to vaccines, looking at the SARS-CoV-2 variants. Because when we go back to the virus, what happened with the virus in our cells, and we look at the spike protein, this virus that we learning about this spike protein, this was essentially the original wild type virus, or the sometimes called the Wuhan virus. This is the SARS-CoV-2, the original strain. Now, all the protein-based vaccines that we discussed and the gene-based vaccine were either I get this gene, I give it to a person, and the person develop the spike protein, or I give the gene and the person develop the spike protein. But these genes were always from this original wild type virus. Now, what we know what happened is that, and this is, everybody went back to school to learn the Greek alphabet when the World Health Organization said, okay, any type of variant for the SARS-CoV-2 is going to have the letter of the alphabet. Then, we have the first variant that was the SARS-CoV-2, the alpha variant. And the alpha variant started in October 2020, described in the United Kingdom. A little bit of what happened then. We know that by at the beginning of 2021, then we have the alpha, beta, gamma, delta. This was the beginning of delta for 2021. It was 1% of the US cases. Then April, May 2020, we started with delta. By September 2021, in a couple of months, 99% of all the US cases were delta. Then the wild type and the alpha type were essentially completely eliminated in a couple of months with delta in the US. Now we know that the story, this is September 2021. We know that in December 2021, the new player, the Omicron, that started in South Africa. And is the end of 2021, and we are already, Omicron was 55% of the US cases. And we know that by January 2021, 99% of the US cases were Omicron. What I bring here, the point is that we moved from the wild type to the alpha to delta to Omicron, and now the different subvariants of Omicron. Then the challenge that we have is that every 4 months, 5 months, we've been changing the type of variant that was the predominant variant, and this happened in different countries at different times. Then what happened with the delta on the Omicron? If we go back to, we received the vaccine with the original wild type, and we have the optimal immune response with good amount of neutralizing antibodies for the wild type. Okay, this is the way that we're supposed to look at delta and Omicron. Now, with delta, the neutralizing antibodies that was produced for the wild type, they still work, but they work less. For Omicron, again, it's another step higher. They still work, but they work less. And we still have all the vaccines going around, still produced with a wild type. Then what's been our answer to this Delta Omicron? Well, when you are a hammer, everything looks like a nail. Well, we just come out, well, let's give another booster. Let's give another booster of the original wild type, because this is the only thing that we have, hoping that the booster is going to, well, hoping no. The booster always give us a little bit of response. The question is for how long this, when the waning immunity, because we are learning that with every booster, the immunity for this booster decrease. With the first doses were 6 to 9 months. With the second dose, probably 4 to 6 months now with this booster. Data from Israel suggests that probably it's going to be just a couple of months, and we are going to go back again to minimal number of neutralizing antibodies. Then the answer we all discuss is we need to start developing vaccines, not for the wild type, but for the variant-specific vaccines. The challenge is going to be that if this continue, every 6 months we may have a different variant predominant. Another challenge is, we are going to have multiple vaccines. Probably going to be 5, 10 different vaccines approved in different countries. How do I compare the vaccine? What is the benefit risk analysis for all these vaccines? Well, it's going to be almost impossible, because how do we compare because we have the phase 3 clinical trials published in New England Journal, JAMA, in very good articles. And we have this vaccine is 90% effective, the other is 85%. How do we compare this? Well, the problem is that clinical trials, the original clinical trials, were with when the wild type was circulating. Having clinical trials now with Delta and Omicron is really comparing apples and oranges, because a vaccine effectiveness of 95% for the wild type, and now a clinical trial with Delta or Omicron with only 80% effective may be as good as a prior 95%. Different variants are going to have different effectiveness. Another issue is the waning immunity that we need to consider. The methodology of the clinical trials sometimes were done in other countries when the patients were already having COVID first, and then they have a vaccine. You have COVID, you already have some immune response. You get the vaccine after COVID, then this patient is going to have probably, it's almost like having a second dose. You have COVID and you get a vaccine, you already have 2 doses. Then this vaccine is going to look better if some of your study population had prior infection. Another important consideration when we look at the literature is that we need to figure out, what is the outcome? Whether the vaccine works 99%, 80%, 70%, against what? Because against mortality, seems to be that most of the vaccines are good. Most of the vaccines prevent mortality. Against hospitalization is different, but against disease and against infection, can you prevent disease? And almost all the vaccines have been a problem to prevent infection. Then you still can get people that are going to be infected but maybe asymptomatic. And this is a challenge, because a vaccine that prevents disease is very good for a person. From an epidemiological perspective, you need to prevent infection to prevent transmission. Finally, adverse events. We know that the viral vector vaccines, we have the thrombosis with thrombocytopenia syndrome. We're not going to go into detail of this. We know that it is an autoimmune response for antibodies against platelet factor 4. Because of this, just a couple of weeks ago, the CDC, the FDA mentioned that at this moment, at least in the United States, the Johnson & Johnson vaccine essentially is supposed to be used only when you cannot use the other vaccines that are available, primarily the Pfizer or Moderna vaccine. Then essentially the Johnson & Johnson vaccine, for practical purposes, is not going to be used anymore in the United States. And then for the mRNA vaccines, we have the myocarditis, pericarditis issue. Then essentially 30 minutes, I need to stop. This is, of course, it's a tremendously large topic. We discussed immune response to natural infection, that we need to understand this better to see what's going to happen with vaccines. And now we know that natural infection confers some form of protection. Protein-based vaccine, the traditional approach, the way that we've been doing vaccines for a long time. We have at this moment approved inactivated viral vaccine. There are still research in other type of, for instance, the attenuated SARS-CoV-2 vaccines, but there's no phase 3 of this vaccine. Protein-based vaccine, the gene-based vaccine also approved in multiple countries. The immune response to vaccine, the idea of neutralizing antibodies that we can measure in the laboratory. And then we give a brief overview of the challenge with the COVID variants that keep emerging. because as we know, we are going to be able to control this pandemic when everybody all over the world is vaccinated. And for this, we're hoping that a high production of protein-based vaccines is going to resolve of the massive vaccination that needs to happen in low-income countries to prevent probably keep the emergency or new COVID virus. With this, thank you for your attention. Ruth, we are open for comments or questions.
Okay, good. Thank you very much. While you were speaking, I put some additional information in the chat. If you can open that and take a look, there's information from the Immunization Action Coalition with some Q&A documents, also a new link that CDC launched that can help people make some decisions about booster doses. You may get questions as a healthcare worker, your friends are going to ask you, or those conversations at church or other places. So having information so that you can keep up will be important. Now, one of the questions that I have, if people have questions, please unmute and ask them or put them in the chat. But I know that we have a lot of questions now from people about, whether or not I should get a booster dose. And you talked about that, and you talked about the short duration. At least what we think is a relatively short duration, to me, surprisingly short duration for the vaccine. And then what that may mean with Moderna getting ready to release in the fall, they'll have an updated vaccine that is should be, it is targeted to address more of the variants. What are some thoughts that you have on how people make decisions, and how do you kind of forecast effectively for yourself and others, other than the crystal ball Ouija board?
Yes, it's a difficult decision. We understand that companies Moderna and Pfizer are working with some of the variants, and the question is for anyone: do I get another booster now? If my prior booster was 6 to 9 months ago, do I get another booster now, or wait to see if in the next 1 or 2, 3 months I may get, instead of a booster, I get a booster, but with a vaccine more that is going to be more effective for the variants. And this is it's almost like I would probably, even though I hate to go against the recommendation at this moment is everybody get a booster. Then just to be clear, but what you alluded, Ruth, is someone low risk may want to wait 1 or 2 months to see if instead of this booster, because then I may not be able to get a booster for another 6 to 9 months, wait for to see if I can get a booster with some more COVID variant specific vaccine. I may say that for immunocompromised individual high risk, we try to protect them as much as possible. If a person is another thing that is also interesting talking about boosters is that now data is emerging that probably having a booster with a different type of vaccine may give you a little more of neutralizing antibodies. Then if you get the primary series with Moderna and a booster or people got the primary series with Johnson & Johnson, and then a booster with Pfizer, and now the second booster probably is better together with Moderna. And then because, and then of course, there are some studies comparing vaccines to see what vaccines, at least in the United States, the only vaccines that we have here, the retrospective studies, that seems to be that the neutralizing antibodies, you have Moderna, Pfizer, and Johnson & Johnson will be the way to run these vaccines. But I'm very concerned with the data from, as you alluded, from Israel, that the second booster was just 2 months of neutralizing antibodies, and then there was nothing. Then I don't know, but maybe sometime I'm tempted to wait to see what happens when we get a more variant-specific vaccine. Having said this, I think that yesterday the FDA approved a booster for pediatrics. And we get all these questions. We get all these questions. One thing that to me is when we say vaccines are safe. Well, it all depends on the definition of safe. Because a vaccine can give you myocarditis or pericarditis, I would not say vaccine is safe. Vaccine is risk-benefit. You have to take the vaccine. Because the risk is always higher than the risk is lower than the benefit. Now you give me full vaccination, I'm going to have a child that is already vaccinated, and now a booster. I need to have data on what is the risk on an adolescent of myocarditis versus, then the risk benefit keep changing as we are. The person have COVID already and is fully vaccinated. And now I'm looking for a booster in a 20-year-old. That is the range that you can get. And now, what do I get? I need to be looking at the data and try to make my decision as a provider. What do I gain for the booster? What do I prevent? Because at the beginning, when anyone was going to the hospital and die of COVID on a ventilator, I mean, risk benefit, there was no question. Everybody should get the vaccine. Even with we say, oh, but the Johnson & Johnson vaccine, we have one death every so many millions of doses. Well, you had COVID, you have much more death than one death. But now, for example, now the FDA say, well, one death out of so many millions is one too many. Because now we have vaccines that have 0 death. Then all of a sudden, the risk benefit, we need to keep playing with the risk benefit.
I think too, as you mentioned, that when we have conversations with patients or others, that we will hear, the rates of serious illness have declined. And clearly, when we look at hospitalizations and so forth, that is true. We still have significant numbers of people who are testing positively in the at-home tests, and we don't have a good, reliable number, but we know disease is still present. And vaccination still plays an important role, but it is hard, especially at this point, to help provide people with information, because I think information fatigue and probably Facebook fatigue and everything else, and that patients, friends, families, and others will ask for advice. And I think listening to your summary that it's risk benefit and that we don't want to get COVID. We don't know who necessarily will have the worst outcomes. So vaccination is one way to protect. It's not the only way, but it's the predominant way. But nothing is without risk. And individuals are going to make individual decisions.
To me, what is interesting is see that we go back, the percentage of Americans that have not been vaccinated. And these are the ones that we see in the hospital under ventilator. And then, of course, the patient that is immunocompromised, that none of the vaccines work. But this is a different topic that will be the topic of the antibodies. And this is a critical component of the management of these patients. But it's interesting to see still the large number of patients, the large number of individuals that are not being vaccinated in the community. Then having individuals unvaccinated that are still on a ventilator in the hospital, this is shocking. But on the other hand, it's good to see that every company doing a phase 3 vaccine for severe COVID, for ARDS COVID, they are not finding patients. The study has been canceled in the US and Europe because there's not enough patients on ventilators to study severe COVID in Europe and the US just because of the vaccine.
Any additional questions or comments? Well, thank you, Dave, for putting information with the link to the YouTube site for recordings, and we will continue to add to that every week. And we will see you then next Wednesday when our topic addresses some of the new AAMI standards. This will be particularly relevant to the infection preventionists and individuals that are involved in the departments in hospitals and outpatient settings that are using the reusable medical devices. So we will see you next Wednesday. And Dr. Ramirez, you have any closing remarks?
No, thank you. Thank you very much for your attention. We'll see you Wednesday.
Thank you.
Thank you.